Uh, Hi, Maxime here. So I'm glad today to welcome you again for another bite-sized talk. Today it will be about the pipeline Sarek, and I think it's a very good opportunity that we have one of our power user who will be presenting this pipeline instead of me. It would be sobering if I were to present that. So I would like to thank again the Chan Zuckerberg initiative to help us do this bite-sized talks every week. And right now, let's head over to you, Jose, to present us Sarek.
Thanks, Maxime. Hi, everyone. I would like to start by thanking the NF Core and the Sarek people for allowing me to give this talk. As Maxime said, I'm one of the power users and now also a contributor. And I would also like to thank NF Core community in general. I'm also an active user of other pipelines, and I'm always present on Slack, and I'm very happy of this initiative and how the community is growing, and they're setting up now the standards for pipelines. So I'm really thankful. This talk has been live streamed and recorded on YouTube. So I'd like to say hi to everyone that is out there watching or will be watching later on. A little bit about me. I guess this is a mandatory slide, but I'm not gonna talk too much about me because this is about Sarek, it's not about me. I have a bachelor degree in computer science, a master in computational biology, and a PhD in computational biology. You want to check something about my research or my projects, you can see my GitHub or my ResearchGate page. And some facts about me: I'm born and raised in Spain. The picture there, right there, is my hometown this summer. But I did most of my studies and my career in Sweden, Stockholm, and I got to meet Maxine and Phil and some NF Core people there. I'm one of the original developers of spatial transcriptomics, which is a technology that you might have heard of. And so I accumulated a bunch of years of experience both in academia and industry. I was even group leader of a bioinformatics unit at some point, but I decided to change careers and move to industry. And now I'm senior consultant at BioLizard, which is a consultancy company that specializes in bioinformatics. But that's enough about me. The first slide that I like, many of you probably wonder where does the name Sarek comes from. So I found out that this comes from a national park in northern Sweden, and this picture is beautiful. So if you have a question about why this national park or why this name, you can ask Maxime later. But Sweden is beautiful. I spent there 10 years. So Sarek is a Nextflow NF Core pipeline to detect germline and somatic mutation in whole genome sequencing and whole exome sequencing data. As you know, the pipeline is written in Nextflow and is part of the NF Core pipelines, and it's heavily used, I think, and the community is growing. I see the Slack channel growing and the amount of users growing. And I'm one of the users. I started getting familiar with Sarek as a user, but now, as I say, I'm also contributing. But the main authors are Maxime and Sylvester. I believe this project started in Sweden, Stockholm, and it was started by Maxime and Sylvester, but then a bunch of contributors have been adding in the last year. I'm one of them, but also Gisela, Frederick, and many others. You can see this in the GitHub page, the list of contributors. In the GitHub page, I say, in the Sarek page in NF Core. I also have to say that this work is published, or at least soon to be published. It's peer review, I believe, at this stage. So you might also want to check the publication. Is public access. Sarek, it's, as I said before, it's a pipeline, Nextflow pipeline for basically doing variant calling in genomic data. This is a really nice illustration that is part of the Sarek documentation on the Sarek GitHub page. As you can see here, Sarek is designed for both germline and somatic workflows. This is the main workflow. It's a very high level, but Sarek does more things. But yeah, most important thing is Sarek follows the J80K best practices, 40.0, which are the standard for the pre-processing of genomic data for variant calling. Sarek, one of the cool things that I like, has a lot of tools, both for Germline and Somatic workflows. For example, for Germline, it has haplotype color, free pages, pileup, Strelka2, Manta, TDIC, I don't know how to pronounce that. And for Somatic, obviously Mutek2, which is very popular, free pages, Strelka2, Manta, ASCA, Control Freak, MSI sensor. But it does more things. This figure is a very high level illustration of the workflow, but Sarek does also quality trimming with Trim Galore, which is a wrapper around cut adapt, QC with FastQC, BankQC, a mapping step, which is essential, is done with BWA or BWA2. As I said before, it follows the J80K for best practices for pre-processing and marking duplicates in the realignment. Also, I'd like to mention that Sarek can be used in tumor-only mode. It's a somatic mode, but if you only have tumor samples, Sarek has been updated so it can work with tumor-only samples. And it's also compatible with exon and targeted data, which is very nice because essentially Sarek contains almost everything. It's originally designed for mouse and human references, but technically possible to use other references. As far as I know, me personally, I haven't used any other reference, but there might be people out there that have used them. And it would be nice to someone can say that at the end. I would like to start by talking about Sarek Germline mode. When one wants to do Germline variant calling, is when we have samples that are not somatic, or they might be somatic. You might want to be Germline variant calling, but it's essentially to detect variants that are not in the reference genome. So let's say you have a sample and you want to see the variants that are not in the reference genome. They could be genomic variants that want to inherit. In order to use Sarek for the Germline mode, you just need to include, because Sarek has this option to allow multiple tools to be run. So if one of the tools that are provided are from the Germline toolset, Sarek will do the Germline workflow. And these tools are haplotype color, Strelka2, free pages for SNPs and indels, Manta for structural variants, and TIDIC, TIDDIT for structural variants. And the input here, the input for Sarek is always a tab delimiter file, which is kind of standard in F-core pipelines. So the user needs just a tab delimiter file with information about the samples and path to the raw data. And the output would be BAM files for the intermediate steps and VCF files for each of the callers that are included in the run. And of course, a report, a very nice report in web format done with MultiQC. Somatic mode. Somatic mode, probably quite standard case for variant calling, is to detect variants using a reference genome, but also another sample, a normal sample. This will be variants that are, let's say, somatic, that are acquired, that might be specific to a tumor, specific to a cell type, or to an individual. For this, we need the tumor sample, but also the normal sample that can be used as a reference. How to run Sarek in somatic mode, just as I said before, to include tools that are for the somatic workflow, like Mutek2, Strelka2, free pages, Manta, ASCAT, Control Freak for copy number variation, and MSI sensor for MSI status. In this occasion, to run Sarek in somatic mode, the input file has to contain both the normal and the tumor sample. It has to contain this information. It has to be indicated which is which I will describe later, and the path to the files. And the output would be BAM file, VCF files, and other files, because, for example, the copy number variation and the MSI status, they don't generate VCF files, they generate other type of files. And as before, the reports in MultiQC. Sarek also supports tumor-only mode. This is a case where you might want to run a workflow in somatic mode, but you don't have a normal sample, you only have a tumor. This happens sometimes. Of course, the variants are less reliable, you are more prone to have false positive, but it's something that Sarek supports. I actually one of the users of this mode. As before, in order to use Sarek somatic mode, tumor only, some of the tools that are supporting the tumor only mode has to be included, like Mutex2, Strelka2, Manta, Control-Fig, MSI sensor, and a tab delimiter file as the other modes, but this time only with the tumor sample. The output will be the same depending on the tools. You always have the BAM files, intermediate files, but depending on the tools that you include in the analysis, you will get VCF files, copy number, MSIs, and the report. I have mentioned before the input file, the tab delimiter file, and this is something that I see that's users, especially when they start, they struggle a little bit with the format of the input file. I did struggle myself. The input file format is a tab delimiter file that has a bunch of columns. The first column is the subject ID. This will be something that would be, it could be also the dataset ID or project. This will be something that will be appended to the output, to the different files, the different folders. The gender, which is used in some of the tools. And the very important column, the third column, is whether the sample is tumor or not. 0 means non-tumor and 1 means tumor. And this is very important for Sarek to know whether to run the somatic mode, the Germline mode, somatic mode in tumor only. The sample ID, of course, this is important, especially this will be appended to the output, but especially when you have multiple lanes and you have multiple FASTQ files that you want to merge for the particular sample, Sarek will take care of that. So for example, in this case, sample ID 1 has 3 different lanes. This will be merged before doing the processing. And the path to the files, of course. Simple as is this, sometimes people get confused, but this is the main required parameter for Sarek, and obviously the tools that one wants to run, as well as the reference genome. One thing that I felt it was nice to explain, and for some of you this might be very obvious if you're familiar with variant calling or with Sarek, but I think it's nice to kind of go through what Sarek does, the information that Sarek provides with different tools, because Sarek has a lot of different tools that can provide different information. So it's good to know, to have an idea. The variant calling usually involves deriving SNPs, indels, and structural variants. And so Sarek has some tools that are specifically designed for this, like Mutex2, haplotype color, Strelka2, and free VHS tool will compute SNPs and indels. And here, this figure is quite representative of what this type of information is. Essentially, we have a reference genome that has a sequence, and you have a sample, you have sequence, a sample, and you have the all the sequence of your sample and a specific region. You might see that there is a nucleotide, a single nucleotide that changes as compared with the reference. So this would be a SNP, but it could be also, a chain could be also an insertion or a deletion. So it would be indel, and represented in the second and the third row here. So this would be, these are variants that are detected. Of course, if you're in Germline mode, you only compare with the reference, but if you're in somatic mode, you will also compare with another sample. So you want to see that this change is not only happening in the reference, but it's also happening in the sample that you use as reference. Structural variants are more like bigger changes, for example, like deletion or insertion of a big portion of the DNA sequence, and there could also be inversions. And the tools that are providing this information in Sarek are Manta and TIDDIT. Other type of information that Sarek can provide are copy number variations, or MSI status. The copy numbers are just a difference in the number of copies of specific genes. Different species will have a predefined number of copies that you should have from your mother, mother and father in case of human. So this is a very, very common analysis in genomics to derive the copy number of a sample, to see which regions have a different copy number. It could be more or less. illustrated here with red and green. And Sarek has integrated 2 different tools for inferring copy number status, copy number variations, region-wise, chromosome-wise, and these tools are ASCAD and control-freq. And another thing that is interesting, especially in cancer, is the MSI status. MSI status, an MSI individual will be an individual, hypermutated individual. They usually have defects in the DNA replication, DNA repair pathways. So these samples are hypermutated. They have a higher number of mutations. And this is good to know, especially in the cancer field. So Sarek use MSI sensor, which is a tool that provides this status, which is a score, 1 score per sample. And this, you can see here, the percentage of MSI, which usually what one does is to put a threshold to define a sample as MSI or not. Now we move to the output, the BCF format. Sure, many of you are familiar with this format. This was a format that was designed to kind of include mutational data, variants, and I personally like it. Some people might not, but it's quite standard now in the field. And I think the current version is 4.3. So most of the tools, I would say, the variant calling tools, they make use of this format. The format is quite flexible. It has a header that contains metadata, meta information. For example, it contains the commands that were run, which is very nice to have a history of what happened in that file, how that file was generated. And also explain all the fields, the format, the info. It gives you an explanation of every field. So if you add extra things to the file, usually you have to include it in the header. So you explain what those new fields mean. And then the body. The body contains the mutational information. The first column is the chromosome. Then the position in the reference where that mutation is detected. An ID. This usually is a RS number that is used to identify, to track this mutation in databases, for example. The reference of the genome, and the alter, the new, let's say the mutated, the new nucleotide in case of single nucleotide mutation. The quality of the mutation, the filter. Filter is something that many variant callers provide. They have some kind of confidence scores. They use sometimes probabilistic methods to give confidence to that variance. So some tools, they provide this filter, pass or not, which can be used to process or post-filter the VCF file. Information, this field is quite open. Here, the information about the fields that could be added to the variant, information about the variant, annotation. The format also contains information about the variant, the number of reads in each allele. And then one thing that I like about VCF files is that you can have multiple samples. So the format will detect the information of each sample that is included, and then there will be the samples will be concatenated here. And so one single VCF file can contain information about multiple samples. I don't think I need to explain this here, but of course, in the VCF format or the VCF file, different type of mutation will be represented differently, like SNPs, insertions, deletions, replacement, or structural variants. And if you want to learn more about this format, there's a really nice specification, this PDF here in the SAMTools website. And this picture was taken from this website. One of the things that Sarek does is, as you could see in the workflow, is the annotation, which is something crucial in my opinion. Once you have a VCF file with the variants, you want to annotate them with which to assign the functionality, get the extra information about the variants. Sarek currently supports 2 different annotation tools, VEP, which is developed by Ensemble people. I think it's Variant Effect Predictor, stands for that. Also SNP-F, which I will describe later. Annovar is not currently supported, and I'm not sure that there are plans to include it, but I think it's a proprietary software, so that might make it difficult. But VEP and SNP-F are similar in the way the information they provide. They have differences that some of BP can provide some information that SNPF can provide, and the other way around. But the most important information, I mean, the annotation will create a new VCF file that contains the annotation for each of the variant. And annotations that are quite important is the gene, the transcript where this variant is detected, the feature type. It could even give a consequence. It could tell what kind of consequence that variant can have upstream. The position in the genome, the position, the amino acids change, the codon. It could even predict effects. This is very useful for cancer. It could even use databases like Cosmic to tell you that variant has been detected. Population information, how common is that variant in the population. So this information is quite useful, especially to filter variants or to detect variants that could be potentially interesting. SNPF is another tool. I particularly like this one. It has a really nice documentation. It's really easy to use and fast, and provides similar information: feature type, feature ID, the gene name, biotype, what kind of, is it non-coding gene, is it protein coding, RNA, the impact of the variant. This is something that is specific to SNPF. It has different category like high, low, moderate. This could be used to filter, and also they call it here put the impact, but it's like the effect, the downstream effect of the variant. Once again, I have personally used this a lot in order to filter the data and to detect potentially interesting variants. And the last step of Sarek is output. Obviously, the output is not only the VCF files and the BAM files. It's this really nice report. Sarek will automatically generate a work report. MultiQC, which is standard in the NF Core community. This is a tool that was developed in Stockholm, Sweden by Phil. And I love it. I really like it. It's a web report. It's an HTML report that contains a lot of different stats for the different steps of the pipeline. I cannot show everything here, but yeah, essentially you get information about the mapping quality, duplicates, the type of variants detected, amount of variants, the regions where the most variants were detected. It's a lot of information that is included here. It's quite useful to make an assessment of the data, an initial assessment, to detect samples that might be outliers, samples that might be discarded, and to get a general overview of the data. One use case, I think I have personally used Sarek in 3 projects, at least that I remember now. I think the biggest one was this project that I executed where I was working in Waldebron Institute of Oncology as a group leader, the bioinformatics unit. In this project, we wanted to analyze 141 whole exome sequencing samples. That's quite a lot. And we used Amazon for that, and NF Tower, Nextflow Tower, so Amazon Batch for distributed computing, distributed processing, which is really nice because you can process a lot of samples in a relatively short time. I think it took days once we had everything set up. What we did here was to process different samples. We have germline samples, we have tumor samples. Some of them were solid tumors, some of them were cell-free DNA, and we also have PDX samples where obviously we had to extract the human reads. But all these different samples were processed with Sarek. We use these colors that are shown here in this figure, most of the tools, and we use annotation provided by SNPF, and then we manually annotated with ANNOVAR. We were quite happy with the result with Sarek, and this was one of the reasons why I started to contribute it, because some of the samples here were tumor only, and some of the features that we needed were not present in Sarek, so we just contributed. And that's the best thing of open source. And this project is under the manuscript under preparation. We got really nice results, and we were really happy with the short time where we could process all the samples, the amount of information provided, and the quality of the variants. And this is something very important when doing variant calling. It's quite tempting to just develop your own pipeline, but these tools have a lot of different settings, and it can be tricky to optimize and to run all the steps properly. And Sarek is a pipeline that is built from a community, and it has a lot of users. So I really recommend using Sarek if you want to do genomic variant calling. So yeah, approaching the end. What is next? Sarek 3.0 is coming soon, so stay tuned. I hope that this release will happen in early 2022. I'm contributing to this release, and I'm quite happy to. One of the highlights of this new release is that it will be ported to DSL2, which is now the new syntax, the new language for Nextflow. It will include more tools. I think for now, deep variant. I might be missing something. Maybe Maxine later can elaborate here. It's been refactored and redesigned, obviously, because DSL2 it forces you to do that. But now Sarek is more modular. It has a simplified design. It's easier to navigate it. It was a bit difficult before. And more tests will be added. The testing will be improved, and validation tests will be included. The joint variant calling, which is something very useful especially when using the Germline, Sarek Germline mode. And downstream analysis, this will be really nice, maybe to add the option to filter VCF files according to certain criteria, maybe merging. This is something that is now ongoing, and it might be very handy for users. And decrease the resource needed. Sarek, the current version of Sarek is doing computation that are not needed in certain cases. So Sarek now will be optimized, so only what is needed will be used. And so get involved. I recommend that if you're a user of Sarek and you like programming and Nextflow, if you have something that is missing, I really recommend to get in contact with the community. There is a Slack channel, and people are quite responsive and very helpful, very nice. There's also a GitHub page where you can see instructions on how to contribute. So I really recommend that you're using Sarek and something is missing, just at least say in Slack channel, and you might get it. You might see it happening. So last slide, the acknowledgements. As I understand, Sarek was initiated at Karolinska Zeiss Life Lab, which is a place that I know very well. I spent there 10 years. And so thanks to these institutions for supporting. And obviously, NF Core community, I say many times, and personally quite thankful for all the effort that I am doing. Nextflow Tower, BioLizard, which is the company that I'm working for now, and they're supporting my work now with Sarek with the 3.0 release. The Balderon Institute of Oncology, which is a place that I was working where I ran that project that I explained before, and I did some contribution to Sarek. And QBIC, which I think is also a contributor, I believe Gisela, and they are also supporting the development on Sarek. I think that's it.
Yes, thank you very much, José, for the talk. We have a couple of questions already. I see that, okay, Lawrence has a question, but you already responded to it. James has a question about, like, in the current Sarek version, does mutex2 support multi-sample calling? So I don't think it does in the current version, but definitely that's something that would be interesting. So we will have a look. Maybe in the 3.0 or maybe like in another version. So that's something we can think of. And then another question from Lawrence again. Do you have any idea how a snip effect defines the putative impact?
I cannot hear you well. Sorry, can you repeat?
Yes, sorry. Do you know how snipf defines the putative impact?
I think they have some database.
Yes, I think as well they have some database about that, but
I just had to say that you have to be careful with that, because sometimes, for example, moderate variants sometimes can be quite interesting. So how a snip effect defines these different categories, you have to be careful, because sometimes missense mutations are defined as moderates, and I particularly in one project that I had, I was filtering out moderate variants, and I realized that they were the most important. So one is to use these filters, has to be careful but essentially the filter is just including a bunch of effects. Which effects are included in this category? You can see in the website. And they have documentation. It's quite nice.
Yes, I think Laurence has a question about like modifier and moderate. So yes, she will double check the documentation. Okay, no, thank you very much for the presentation. It was like super clear and everything. But okay, I might be a bit biased about that, but definitely I will steal you for some slide like for my own presentation, because that was like even clearer than what I usually like explain stuff. So that's good.
Yeah, thanks to you, I mean, and everyone attending. Okay, then I think we are good there. Then thank you very much, and see you another time. Yeah.